Okay, I really am honored that you guys are here, not just at 4 o'clock, but at 4 o'clock on a Friday. So I'll try to talk fast so you won't get completely overwhelmed with having to sit and listen. What I'll be talking about today is, let's see, here it is. Is this a pointer? Oh, there it is, okay. Is extracellular traps in plant and animal defense, new insights into mechanisms of eukaryotic immunity, and amoeba have been added to the list in the last year. So it's continuing to spread among the creatures. What I'm going to do today is first briefly summarize where the term extracellular traps came in and how we got trapped into it. But I'm first going to summarize what that is very briefly, and I will be happy to answer questions about traps and cancer and Alzheimer's, etc. But today I'll mainly be talking about the plant side. I do have a few hard copies if anybody would like to have them scrutinized while I'm talking. And I have a hard copy of, got it, yay! So the term extracellular trapping was introduced in 2004 when Arturo Zaklinski's lab discovered for the first time that neutrophils function in a way that's completely different from what we thought. And that, what he found is that they actually export a gooey matrix which functions as a trap, which then allows the neutrophil to kill the pathogens with the various antimicrobial proteins and other metabolites that are there. And they called them extracellular traps, and they found that the central core of the trap is DNA. Totally against the rules, of course. They were able to show that, yes, DNA is there, and if you get rid of it by adding DNAs, microorganisms can get free and cause disease. This was followed rapidly by a paper in which extracellular DNA's knockout in group A strep was carried out, and it was found that only knocking out a single extracellular DNAs eliminated the ability to eat flesh. And between 2006 and 2009, in addition to the plants, it was also became clear that it's not just bacterial pathogens, but also fungi, protozoans that function using being trapped by this mechanism. And since 2010, the work has progressed in defining the other side of the double-edged sword, as one of the reviewers put it, which is that the presence of DNA functions as a trap very effectively to control spread of pathogens in the bloodstream. The downside is it's a very sticky matrix, and if there's not adequate clearance, it can cause big problems. As a colleague at Harvard put it, Denise Wagner, she's pointed out that the traps serve as a seeding soil for cancer. And again, I'll be happy to talk about it. Since that time came out, it's now been implicated in pretty much all aspects of immune and autoimmune responses. For example, lupus, actually there's mutations in DNase 1, and that's what causes the problem. Very subtle. But there have been this stuff for a long time that we've overlooked because we had rules about what DNA was supposed to do, and this didn't fit. And now it's not only infectious diseases, but MS, infertility, Alzheimer's, sepsis, cystic fibrosis, and Alzheimer's. So again, I'm happy to answer questions about that, but you need to let me know because I have to put my helmet on so my head doesn't explode when I talk about human diseases. There's a reason why I went into plant diseases. Okay, so what I'll do now is I'm going to give is that me banging on this that keeps banging? Okay, so what I'm going to do now, I also have a copy of a snapshot article that Fushi and I wrote with Amato Kuzo over at the Cancer Center, which also I've got a couple copies of that if you'd like to see it. What I'm going to talk about today is the function of root border cells in plant health, what they are, why we call them that, and how they got us trapped in looking at the same responses as functioning in humans. Now, border cells are the cells that have always been called sloughed root cap cells. This is one day's number of border cells. This is a legume root. And these have all traditionally been called sloughed root cap cells and haven't been paid much attention, even though there's a lot of material here. And Wynn Silk did some direct measurements and found that 95% of the carbon-based material in the rhizosphere is the cells and their associated extracellular material. And even more shocking, Lynch and Witz showed a number of years ago that the amount of carbon needed to make these cells comprises from 40 to 90% of the carbon fixed above ground on a daily basis. So it makes sense to try to figure out why the plant would waste so much energy on cells that were always assumed to be not very interesting. This is a paper by Rogers from 1942, which nicely summarizes the situation that early recognition of sloughing away of root tissues as a normal process during root growth led to little more than speculation as to what the role of such a process might be, and the process was assigned quite summarily, it seems, the function of lubricating the advancing root tip as it forced its way into the soil. I got interested in the cells totally by accident. Is that me? What? Okay. Okay. So holding completely still won't help. Okay. I'll try to remain calm. Okay, so where I got stuck looking at the cells is, as a graduate student, I was using a root elongation inhibition assay with host-specific toxins. And in the course of it, I started looking at these cells, which the cells that are produced here are made by a dedicated meristem in the root tip that is regulated completely independently of the apical meristem. And it's programmed to make a species-specific number of cells to surround the tip, and as long as it's there, it doesn't make any more. As soon as the cells are removed, mitosis, literally you can see mitosis in 5 minutes, and new cells start coming off the tip. And I always like to take a moment at this point in defense of my colleagues who have done so much work on root rhizosphere biology and largely overlooked the cells, is the fact that unless you really know what you're looking for and are very careful to look for them, it's very easy to not notice that they're there. And by that I mean, this is a picture of a root at 99% humidity. Boring little root tip, right? And I should mention at this point, the first thing we teach in plant pathology class is that in the absence of a continuous film of free water, there is no movement of bacteria or fungi through the soil to cause any problem. As soon as there is a continuous film of water, there is potential for trouble. So let me show you what happens to the root tip. And if you look at it under a scanning EM, you can see actually there's piles of stuff on there. It looks like worms. If you put it into water, literally within seconds, the surrounding mucilage gets wet, the cells expand away from it, and if you give it a quick squirt, or if you sneeze while walking across the lab carrying the tray, all the cells are gone, and it's a boring little root tip again. So easy to see why nobody paid much attention to these in any detail. And the only reason I did was because I was using the microscope to look at the effects of the toxins on the root growth and development. And I kept seeing that the cells weren't dead. They were still alive after 2 days and 3 days and 4 days. And of course, I'm the only female in the plant pathology department in Kentucky, so, you know, I felt the need to do it at least 9,000 times before I admitted what I was seeing here. But this is a stain called fluorescein diacetate, which only accumulates inside living cells. And what I found is that the cells of pea and corn could remain 100% viable, literally for weeks, just in water. And they actually could be induced to divide and grow into callus. So at that point, I thought, what is going on? And, God, so I'm too close over here. Is that what it is? Okay. That's the only pocket that I've got, though.
No, I have on your collar. Slide down this piece. I have the microphone.
Oh, there it is.
I put it on.
Don't tell anybody. I couldn't tell what he was talking about. Good, we can leave that. Okay, yay. All right, let's try that. Okay, so I was interested in it, but even more than that, I couldn't understand how I could possibly be seeing this. And this is the 1970s, so looking anything up, there were no Google searches. It was spending weeks in the library looking up hard copies. And I finally found a paper by Leon Knudsen from 1919, who was at the Cornell Experiment Station, who felt obligated to point out that one can find in various texts the statement that the root cap cells of plants die and are sloughed off, and it is probably the general opinion that the cells are either dead when they're sloughed off or they die soon after, that the root cap cells are not dead or short-lived, but may persist for many days. 3 months is what he found. 100% viable after 3 months. Seems to be substantiated by various observations with a number of plants. So I felt validated. But most of all, I felt excited about the idea. The problem that we have, all kinds of diseases, plant diseases as well as the other, is how do we find out what's happening? How can you tell? Once the disease is set in, you don't really know, how do we stop it? And same thing with plant diseases. And what we thought is, these are populations of single cells that reflect the genotype of the whole plant. They have lignified cell walls, and they potentially could serve as a model system to catch the burglar in the act of invading, which obviously greatly increases the chances that you're going to be able to understand what's going on than if you have to do a lot of DNA sequencing and so forth. And the work looked very promising for a while in the sense that you could isolate the cells, get a new batch coming off, and literally you can get a population of cells, brand new population of cells, anytime you remove the existing ones. Bob Sherwood, a colleague, read a couple of my papers and followed up on it by checking, and wow, this is a corn cell, root cap cell, and this is a fungal pathogen called Colicotrichum graminicola, and he was able to show that, yes, you can watch the infection of single cells, and there was a difference between a virulent strain and a non-pathogenic strain interaction with regard to the ability to invade the pathogen, to invade the cell. And I always like to take a moment here to say what a beautiful picture this is of the main issue, which is this is a 1 micron thick lignified cell wall, very tightly contained, and I always say it's hard for me to understand how humans could have decided we're superior when we don't have any lignified cell walls at all, but I try not to say it in company. So anyway, my main focus was with Agrobacterium tumefaciens, and I was interested in trying to see if we could track the delivery of the genes that are necessary to induce tumors in plants. And this is an example of the kind of thing I saw. There was chemotaxis of the pathogenic bacteria to the cells of susceptible hosts. Oops. And there's a tumor, pretty nasty looking. And what I found, very excited about the idea that there was a very strong correlation between the ability of bacteria to cause the tumors and the ability to be attracted and bind to the detached cells. So it looked like I was going to be right. This is an example. This is a group of cells. I mixed them together with the bacterium, and the agrobacterium was hemotactically attractive. They glommed on in huge numbers, looked very ferocious, and taken out of suspension were aggregated together in a mass, which is what you'd expect in order for a tumor to be able to be incited. Got some good pictures. And I'll quickly finish this up and move on to the next stage by saying that I kept getting little hints that my basic premise, which is that these cells reflect the personality, the genotype, the susceptibility of the host plant. So we can trust that what we see with them is going to tell us how disease occurs in the rest of the plant. But I kept seeing things like this particular one is a plant called Erythrorhizum lithospermum, and it makes a red pigmented material called chiconin. And this is probably the prettiest picture we have of this annoying phenomenon, in which case border cells had a phenotype that was completely different from the rest of the plant with regard to expression of the pigment, including the root cap cells that it was a part of minutes before. And I'm going to show a couple of movies now, which they're 30 years old, but they still reflect the dynamics and complexity and drama of what's going on at the root rhizosphere interface, even in distilled water without anything other than one pathogen and one host. Got it? Okay, go for it. Now, what you're going to see here is these are zoospores. These are Mike Stanglini's zoospore petting zoo, was willing to share. If you put the zoospores into distilled water, they'll swim in a synchronized fashion for weeks, unless you put a host plant tissue in there. And this is the assistant professor dragging the root across the slide, getting it into focus. And I'll just let you look for a second, see if anybody notices anything. This is real time. You can see the cells are starting to take up water, but barely. It's literally a few seconds. There you go. I always think I should have the soundtrack to Jaws. Yeah. And the main thing is this is real. And the stunning part was not only was there chemotaxis toward the root, but the zoospores actually amassed following the trail where I had dragged the root across. Instantaneous recognition of something that got them very excited. And now obviously one possibility would be that, okay, maybe there's something leaking from the root and the cells are just in the way. So we washed off the cells, put them into the zoospores. No chemotaxis, no response, no swarming toward the root. And the next slide I like to show just to illustrate not just the dynamics, but the magnitude of this response, which is quite stunning. Number, yeah, video too. Yeah, got it. Okay, what this is looking at the same root at a higher magnification. And what you can see here is, again, the swarming. Some of the zoospores have already encysted. The cells have started to separate away. But I'll just let you watch for just a second over here.
Part of the flow coming from right to left, are those bacteria?
Zoospores. Zoospores. It's an oomycete, Pythium disoticum on cotton. And I'm going to ask that because that's the most important point. What I wanted to say there is just you could see here, which is the magnitude of the material that's being deposited into the rhizosphere. Now we can go on to the next one. So the next question, we wash off these cells, do we still get any attraction to the root? No, no attraction to the root. But what if we squirt the cells into the liquid? Number 3. Yeah. And that'll be the last one, I promise. Okay, so here we squirted the cells. You can see the nuclei. Again, real time, and I have no idea what the signal is and why it goes in the manner that you can see. It's one cell at a time. It kind of looks like lakes in Idaho. You know, they all go to the same spot. Okay. Got it. So anyway, this is what we see. All of the plant cells will be invaded and killed within a matter of an hour or two, but they do it one group at a time. But here's the part that was shocking. So this is a species Exoticum, which is a host, which is a pathogen of cotton. And that's what we see. If we take the same pathogen and put it on cucumber, absolutely no chemotaxis, no response, no attacking whatsoever. Conversely, the pythium that infects cucumber, no response to the cotton. So what this told us that all of my presumptions were wrong. These cells are not just like the rest of the plant. They have a different phenotype even than the cells they came from. And we needed to take a look at what in the world was going on. So the next slide, very briefly, I just want to say that we looked at a number of different bacterial, fungal, zoospore, nematode pathogens and saw the same thing with all of them. There is specific recognition of the cells, and what appears to happen is that they're rapidly stimulated to germinate, and then they are anesthetized and put down for their nap. Most of them aren't killed. They can come back to life after 5 days or so. But by then, the vulnerable part of the root is gone, and there's not much they can do. And I'm going to briefly mention that one of the things we also did was found that the trapping metals as well. Aluminum, in response to aluminum, the cells make a big, this area here is revealed within the ink, and that is the DNA matrix. Can't penetrate into it. And I'll say more about them. I'm going to say that for the end parts. But the bottom line is, I realized we really need to see what in the world is going on with these cells that make them behave in a way that's so different from the others. This was the mid-90s, so we used 2D gels, put them onto filter paper containing radioactive label, and then wash the cells off in the liquid and run the 2D gels. and I should say that there was some talk during that period, and he said, now I'm going to show 2D gels, and I understand that 2D gels are like children. You can never get tired of looking at all the similarities and fascinating differences in your own, that everybody else's look just alike. But I would submit that even if it's not your gels, oops, apparently I need to go faster. Even if it's not yours, the profile of the newly, this is a 1 hour period. So protein synthesized in a 1 hour period. And this is the cells made by the root tip, and these are the ones from the border cells. Completely different profile. There are about 118 proteins. And the really shocking part is almost all of them. So it was at this point that I realized we were in sloughed root cap cells, because they're not really sloughed root cap cells. First of all, they're totally different from the root cap, so it's wrong to call them root cap cells. But I grew up on a cattle farm, so I hate branding and name changes and all that stuff. But I finally gave in to the concept when I finally looked in a thesaurus and discovered that the only synonyms so I had been asking NSF for funding to study infection with putrid gangrenous cells. And we chose border cells in honor of Nogales, which officially is a part of Mexico, and it's officially a part of the U.S., but it has a very special personality on its own, and that's a good reflection of these cells. Yes, they're part of the rhizosphere, and they're part of the root, but they're very different. So we set out just to simply test predictions of the hypothesis that these cells are as a decoy to trick the pathogen down so that it can't harm the root tip, which, as Darwin pointed out, you can mess with almost any part of the plant. If you mess with the brainstem, you're in trouble. So we set out to, first of all, just a couple pictures to illustrate the basic concept, which is that, can everybody hear me okay? Okay, good. This is an illustration of what normally happens in beneficial as well as pathogenic interactions. This is using pouches so that we can watch it happen as it happens. The arrow indicates where the root tip was at the time we added the inoculum. This one has been infected. It's a little teeny bit yellow, where infection was initiated in the region of elongation where the cells are not lignified. But in most cases, most pathogens can survive as endophytes for a significant period of the growing season without really causing any problem. And this is a control cell, control root here. No effect on the growth of the root. This is another example. This is a pathogen called FOMA, which makes a pretty significant lesion. Again, the control, and there's a significant brown lesion that developed, but it's still not having any effect on growth and development. Unless, in this case, only about 1% of the time does it actually hit the tip. And when it does, done for. Even though it's not rotted, that root's done for, and most of the plant too. If you look at it under a microscope, you can see the fungus actually grows into a fuzzy mantle, covers the root, and if you put it into water, it peels off, and literally, this is sterile. I wouldn't even thought of doing it. One of my students said, let's see if anything grows out. Across nature, something's going to grow out of it, but it was not. It was up here. And the mantle rolls back into the rhizosphere. If you look at higher magnification, you can see in every case the cell is enmeshed in it. And this is just the numbers. Very quickly finish up here to show that if you just put the fungus into root exudates, they grow in a linear fashion over days. If you put them into the presence of the border cells, they germinate very quickly, and then they flatline and they don't grow anymore. Yeah. Okay, so obviously, next step, and I'll very quickly run through this and get to the main point, which is to finish up with figuring out what was going on, and then a couple examples of work that we're doing now to explore the possibility of putting this to work in order to have healthier plants and cleaner soils. So obviously the goal at this point was we need to do some knockouts and make some mutations in genes in the root tip that we can use to control the production and the properties of the cells as they come off, to see if we can identify what is the signal, what is it that allows it to recognize it, how does it shut the pathogen down. And to make a long story short, we succeeded in doing a lot of knockouts, but of course it was using cell wall, cell division pathways, pectin, cell wall breakdown pathways, and yes, we got mutants, and yes, there were alterations, but the effects were pleiotropic, change in pH of the cell wall, etc. So we really couldn't use those to draw any conclusions about how it worked. And at that point, we thought, we really need to go back and look at those stupid proteins and figure out what they are and see if they give us any clues. So we did, and again, 118, and they were, most of them were predictable antimicrobial stress proteins involved in defense pathways, carbohydrate, cell wall metabolizing, proteases, all made total sense, except for one, histone. And it's like histone's inside the cell holding our chromosomes together. There's no way it's going to be outside the cell, right? So we did a lot of controls and essentially, to make a long story short, found that if the cell isn't alive, nothing goes in or out. So we could add secretion inhibitors, we could kill with heat or cold, and no proteins leaked out. So luckily Fushi was keeping track of the literature, and she said, you know, there was a paper that just came out about histone outside of the cell, neutrophils, and we've been trapped ever since. It's your fault, Fushi. So this is a paper from Zaklinski's lab, and again, they found there was this fibrous material that the pathogens were attracted to, and they aggregated, and when they checked, they found the DNA was a component of them, and that they added DNAs, the pathogens swam away, and were unable to control the infection. And this is my personal favorite. This is a study that was done in 2014 in which they followed up on the mutations and all that to ask when, where, and how did group A strep start eating flesh. And the bottom line is they sequenced 3,615 strains from 7 different countries, traced it back to the 30s, and identified the time and place when it went from being just another pathogen that didn't cause much trouble to eating flesh. And what they found was that what happened was that it contained extracellular DNAs. Yeah. So what in the world was going on? So we went back and looked, and we followed in Arturo's footsteps and did the same thing they did, which is, seriously, DNA out there? Obviously, the DNA strands look very much like the neutrophils, very clear. And it made sense now when I thought these were ferociously attacking the cells, that actually the cells were ferociously attacking the microorganisms. Again, this is an image, again, to remind you the magnitude of the slime layer that's out there. And again, dynamics of the rhizosphere, a little overwhelming. Okay, not my fault? Yay! Okay. Okay. It probably doesn't want me talking about this stuff. That's what it is. So anyway, we already knew the little layer that looked like biofilms, and I checked to see, and sure enough, you can use India ink as with biofilms to see the magnitude of. And this is a corn one, same thing. So this is, the cells separate, they have one of these little layers. What happens within about 10 minutes after adding a pathogen. It expands dramatically, and you can see the bacteria trapped inside. So Fushi proceeded to use DNA stains to confirm that yes, DNA was present outside the cell. This is using DAPI. This is also DAPI, which goes inside the cell, so it's not too specific, but it allows you to see the magnitude of the trap and all the bacteria that are there, which I think is always important when I read the rhizosphere microbiome, trying to make sense of the distribution. It's going to be really important to notice that this kind of thing is going on with localizing the pathogens. And this is Cytox green, which only stains the extracellular DNA. And the first thing I thought was, this is completely crazy. Some of the elegant work I've seen done in science is by my colleagues working on characterizing the mucilage surrounding these cells. I just believed there could have been DNA there and they wouldn't have noticed. So I called them up and they all very cheerfully said, oh, and I said, what are the chances that DNA could have been there and you wouldn't have noticed? And they said, oh, nope, absolutely. The first thing we do before we do an analysis is treat with DNAs to get rid of the contaminating DNA. Okay. And as it happened, it turned out there was a paper in the 68 and 70 by a couple who have used root caps as a model to study cell cycle. And you want to get up and walk around, that's okay. I can't sit very long. You can join me up here. But anyway, what they found, to their shock, was this just shows the meristem, and you can see the increased amount of DNA being synthesized in there. Makes a beautiful model to study cell cycle because it's very easy to visualize it. But what they found is that there's as much DNA being synthesized at the cap periphery as there is in the meristem. And was parts, it would have been so easy to just chop it off and not mention it, but they both published it, said, we have no idea what in the world is going on here. But the next thing we did, again, following Burroughs' footsteps, is to say, okay, what if we get rid of it? And in a nutshell, this is a plant that's been infected. It's growing fine, no sign of infection. This was one where the DNase was added at the time of the inoculation. And we go from 0 root rot to 100% root rot overnight. DNase, and this is a confocal picture of the moat around the castle. You can see there's literally an area where the microorganism can't enter. This is a fungus. The hyphae is truncated and not growing much. Occasionally it'll stick its nose into the root tip, and the response of the root tap is to start pouring border cells off. And even in cases where it makes a teeny little lesion, it peels off. This is what DNase, the fungus proliferates madly. The poor little root cap is lots and lots of border cells, but it's not doing any good because the DNase is breaking down the trap. This is just a quick series showing the dynamics of the process. John Kessler took some pictures for us, and this is a single cell. Within 20 minutes, there's a mass around it. Within 40 minutes, a huge mass. This cell is still alive. You can see the nucleus. And the next slide shows what happens when you add DNase. And it's still alive. Within another 20 minutes or so, makes a new trap and gets some more. So start to picture this going on in our bloodstream. You can see the dynamics and the potential for issues with regard to overtrapping causing problems. Now, briefly summarize, or the next step we did, was knockouts, and I asked two colleagues who work on the only two pathogens that I've ever seen that have the ability to cause some damage to the root tip. It's not a whole lot, it doesn't rot it, but you can see the root tip behaving in weird ways. And one of them was Ralstonia solanaceae, and Caitlin Allen at Wisconsin was the one who was willing to do it. And I assumed we'd have to run the proteins out and sequence them and start from scratch. But no, they just did the databases and found there were two putative xDNAs encoding genes in the genome. One of them didn't have much effect. This is wild type breakdown of DNA, and that's one of the DNAs, putative DNAs. The other one completely stopped breakdown of the DNA. And this is what the wild type looked like in response to the host border cells. Not much interest. The next slide shows what it looks like within a couple of minutes of adding the strain with the DNA is knocked out. I don't know if it's statistically significant or not, but but here it is in the whole plant. So this is water, and again, this is what it looks like with the wild type. Significantly inhibits growth, but it doesn't rot it or anything. And this is the nuke A, which didn't have much effect on the DNA's breakdown. The nuke B, knockout single xDNAs, and it's almost back to wild type. And I'm sure you know plant pathology like humans, you don't knock out a single gene and get this kind of changes. You knock out pathways and chromosomes. And this just shows the numbers, even more important, just like with group A strep. So this shows the wild type, and you can see the growth of the bacteria moving through the vascular system. So this is 4 centimeters from the start on both sides. The green represents the presence of the bacteria. The next slide shows what it looked like with the nuke knockout. Almost no spread. And actually, Luis Sequeira, who was also at Wisconsin, was one who pointed out that in most vascular wilts, it's not the microorganism that's clogging the system. It's some stuff that the plant is producing. He didn't know what it was, but he said it was analogous to blood clots and strokes. And I'm just going to mention very briefly now and not get started, but one of the things that has emerged is that if you add DNAs to blood clots, it dissolves them. That's what DNAs in the bloodstream is there for. And the other one, I'll finish up with this one. So this is Cochleobolus heterostrophes, helminthus foraminatus. This is the pathogen that not wiped out 80% of the corn crop in the U.S. in the 70s and was responsible for founding a lot of plant pathology departments. And this is what the wild type, the control looks like. The wild type really damages the root tips. And again, very different from most others. But whatever defense is there, it doesn't work very well against this. She identified a single putative xDNAs, knocked it out, almost back to wild type, and even more dramatic on the actual leaves, which is where it causes the problems. Here's the wild type, and there's the knockout. And the related species, Cochleobolus victoria, there's the wild type, and there's the knockout. Okay, so what are we going to do about this? Well, on the human people, the people who work on humans have been working very hard. We're up to about 2,000 papers on the nets trying to understand what is it, what is the trap, how does it work, how is it exported, how can we understand it sufficiently so that we can facilitate disease control without allowing cancer to develop. And they're just a teeny little bit jealous about the plants. Here's a quote from Arturo's lab a couple of years ago summarizing the problems in dealing with human cells, apart from the fact you can't get 100 neutrophils that are all alive in one place. But neutrophils are short-lived, they don't divide, they can't be genetically modified, there are few cell lines, mice are an imperfect model since they do not mimic human neutrophils in function, morphology, or physiology. In vivo, murine neutrophils can be depleted with antibodies, but this is transient as low neutrophil numbers trigger renewed production. On the positive side, neutrophils are abundant in human blood. It's relatively easy to obtain populations allowing short-term experiments. The limitations are that since the experiments can be performed only in vitro, the cells rest on artificial substrates and media. So the bottom line is we're going on 13 years now, and somebody's trying to target DNAs to tumors with nanoparticles, and there's a couple of clinical trials trying to use DNAs to control symptoms, but the bottom line is that mechanistic issues are not moving along quickly. And what I noticed is there's dozens of papers published on utilizing the nets. So it occurred to us that maybe we should do some of that too. So Fushi took a lot of pictures using different DNA stains. I'll briefly finish up at that, and then I'll end with examples of the kind of stuff we'd like to be able to do now that we're beginning to understand what's going on. So this is a root tip stained with a stain called crystal violet, which stains DNA a purplish blue color, so you can see that. And I'm happy to report that we got away with calling them barbed wire looking structures. They do look like barbed wire. It stains pectin pink, and you can see the pectin stain there, so it's not too specific. You can even see it very clearly in black and white. You can see the barbed wire. If you add DNAs, the barbed wire is gone, but the pectin's still there. The next one is Cytox green, which only stains DNA outside the cell. And here we have, there's a cell here which is alive, so there's no stain inside the cell. This is the phantom rising from the ashes, and this is the dragon trap. I think we should probably be able to make t-shirts out of these. And we got some hints where you can see literally strands of DNA coming out of the cells and attaching to the traps. Another picture of that. This is what it looks like if you treat with DNase. So stay tuned on that. The next slide is just another picture of it. And again, there's the massive trap surrounding the cell. This is P, and in P there's usually 5 or 6% of the cells are dead. So you can see here this is taking up the DNA, so it's a dead cell. So maybe the 5% are producing the DNase. Except that this is what it looks like if you kill the cells by heat or freezing. No DNA gets out unless the cell is in control. And this is something that, again, this is something we can do a lot more efficiently with the plants than the humans. So what are we going to do? Well, as it happened, a local anonymous organic grower showed up with an idea about maybe we should take a look at his compost mixture to see what would happen. You sent me the picture. What am I supposed to do? So we tried Tom's compost tea, and we thought, a couple of weeks we'd be able to rule it out or whatever. We've got 2 papers so far. The first one was looking at root disease suppression, and the second one, in some ways even more exciting, is the potential for altering that carbon delivery and freeing up some extra energy for growing tomatoes. And briefly what was found, this is in the compost, which I thought that was likely to cause some physiological problems, but perfectly healthy, growing the peas in compost, no problem. And this is infected with, that's a water control, and that's one that's infected with the pathogen, with the typical infection behind the root tip, but no effect for most of them on the root growth. This is the compost, no problem, perfectly healthy. Ready? This is the compost added at the time of inoculation. 100% protection, not just at the root tip, but in the region of elongation, 0 infection. And what was even more startling is what it turned out to involve is exactly what I've been working with companies for 20 years, trying to figure out a way to wrap the border cells up around the root tip so it could protect the region of elongation too. No luck with that, but the compost knew exactly what to do. You can see here, this is the region of elongation, and the sheath wraps up around and prevents the. And essentially what happens is this is a root tip placed into water. You can see the border cells all dispersing. In the compost, there's no dispersal. So it doesn't need to make any cells because it's already got them there. So the potential for having extra energy is very significant. And again, up to 90% of the day's carbon may not be insignificant. And now, and for just a couple seconds, where'd Rob go? With what we're mainly working, just kidding, what we're mainly working on now is working with John and Rob and Mary Kay, looking at metals in the soil. Now, one of the things people have tried a lot to do is use filtration to clean up the messes that we've made with lead and other toxic chemicals in the soil. And for the most part, the plants decline to participate. Nobody's been able to quite figure out why. And of course, the reason is because the border cells are there to protect the plant. I'm more worried about the plant than the humans. And we did a paper in 2000 in which we were able to show that this is a snap bean root. This is in the absence of aluminum. This is stained with an aluminum stain called lumigallin. The border cells were removed before we put it on there, and a lot of aluminum goes into the root and it stops its growth completely. If the border cells are on there, very little aluminum goes in and it has no effect on the growth. This was a little bit shocking. And this is another picture showing the same basic thing. We were able to show that The cells themselves make a trap, and there's a significantly more trapping with resistant cultivars than sensitive cultivars. And a group followed up with, did exactly the same thing with soybean, but I like their pictures better than ours. So this is an illustration, this is a control, and this is increasing concentrations of aluminum. You get bigger and bigger traps, and the traps in the resistant cultivar are significantly bigger than the ones that take up more. And we a very exciting epiphany. Monica Ramirez gave her seminar when she came here as a professor, and she had been testing arsenic levels in home gardens in Boston to make sure people knew what they were eating. And bottom line is the ultimate double-blind study. She found that species had large numbers of border cells. There was no uptake into the above-ground parts. But in the species that had very low levels or no border cells, there was a lot. So that got us going. Like I said, we're working with them now, initially just to ask how much lead can the border cells trap, and what are the possibilities that we can put this to work, cleaning up some of our messes without destroying the other creatures. So we're in the middle of that in collaboration with Shore River Lab, and I'm very excited about that. David's working on his PhD with it, and that's it. So again, I think you can see the main thing we're mostly focused on is the possibility for using this to develop healthy plants and healthy soils. And these are all the people that we've been working with, and I'll just end with a quote from Charles. And we all agree with him. Oh, yay, I didn't go over. Yes.
Martha, can I say back
Real quick,
Please.
Okay. There's a difference between what we think of as phagocytosis and the role that a leukocyte has, and that is spoken of as a cell membrane, which has a dynamic, and the cells we're looking at here are cell wall, and to the best of my knowledge, they physiologically behave quite differently. And you pointed out that there's a ability of producing materials that will grab on to potential invaders before they even get to the wall, but then they seem to cluster on the outside of the wall. Somehow, I'm not sure exactly how, but some of them get inside and they die. Is that right?
No, they don't get inside.
They don't get inside those at all. So
No.
everything is death outside of these cells.
I mean, with a couple of pathogens, the fungal pathogens,
Okay.
the zoospores, yes, they can infect.
At a later point, you pointed out something that you called analogous to a stroke, and that was the invasion of some materials into the equivalent of the vascular system of the plant itself, killing the plant.
Right.
Now, I'm not sure I understand exactly how the bad guys either get into the plant, or how does this apply to the people who are raising tomatoes or whatever, and they want to have very healthy plants that don't die and don't carry pathogens forward into a food system. Is there something there that I'm missing as far as what you're telling us that works right, and how understanding this helps us to protect the food systems?
Yep. Well, obviously we're in very early stages of trying to figure that out. And the first stage is to figure out what's happening, and that's what we've got going now. As far as the questions you're asking, that's, and again, the parallels with the humans are hard to resist going into, but the bottom line is the vascular system with the plants, it just clogs the system, so the water flow isn't there, and that's what kills it. And in the humans, obviously, the blood flow issue is the same basic thing. As far as the specifics, and this is what they're really working, I've gone back and forth to Berlin several times, and they're trying really hard to get at exactly that. Even the question of how it's delivered. You know, is it cell death? Is it an active process? In ours, the cells have plasma desmata that they control, little holes they could squirt things out of, and then it closes back up again. So we hope to get at that pretty soon, and we hope that that's going to at least give us hints about, like you said, how do we help the plants keep themselves happy, and the human systems. Stay tuned on that one too. So does that answer your question or not?
Yeah, that leads in, and I'm sure there are other questions.
Hi, yeah, Martha, you early on, in fact, the leaky DNA and the DNA matrix and protective qualities, the reactive properties of the matrix might be in mammalian or animal systems as well as plant systems.
Yep.
So, but animal and plant systems are quite different in that plants have the cell wall. And so it would seem that, and I'm just thinking, that a cell wall might retain some of it, might not leak as much as an animal cell. Do you have any evidence of this extracellular DNA produced in the amount of extracellular DNA produced by animal cells versus plant cells?
The actual numbers haven't been done. If you look at the pictures, it looks very equivalent, very massive, large masses of it. And as you said, in the human cells, there's no cell wall. So getting at where it's coming from and how continues to be a big question. Big question, obviously, because we need the nets to survive, but we need them to be, and I'll just say one thing real quickly, just as reflection of this. DNA, I didn't even know DNase 1 was a secreted enzyme until I started all this, but DNase 1 is in the bloodstream at pretty consistent levels in healthy humans, and it's there to clean up the messes once the pathogens are under control. And the nets use almost all the same proteins that the plants use, the reactive oxygen, proteases, etc. But in people with lupus, they have very low levels of DNase in the blood, and that's what the problem is. It's been shown since the 50s that cancer patients have very low levels of DNase and very high levels of DNA, and the obvious possibility of just getting in there, but I mean, even just the basic questions of what can we do to get our DNase levels up. There's one paper out of Sweden showing that a marathon will increase your DNase levels for 24 hours. So good luck with that. But anyway, some of these things are potentially very straightforward, based on obviously what we know about DNA and DNase already. Yeah.
So in your presentation, you showed that putting compost water onto the plant kept the border cells on. I was just curious as to how you applied that, or was it just sitting in compost water?
Just put it into the liquid. And we also did it in sand as well. Same thing, just put it in there, pour it in. It was quite a shock, I can tell you.
Dr. Pat Laura has a question.
So up on that question about the compost, do you know what in the compost is? Aha.
Yeah, actually that was a big shock. Because obviously, who knows? I mean, I thought, aluminum, who knows? Yeah, that was Starbucks coffee grounds, not just any coffee grounds. But what?
And I don't know if you made it with manure, if that would make any difference, but
Yeah.
it's just straight up compost.
Yeah, but we did look to see if we filtered it, there was absolutely no infection. In fact, the infection was worse. And Gilberto wanted to check and see if there was some species there that we could check on. And I thought, compost, there's going to be 9,000 species in there. It was a single culture. It was a Acinetobacter baumannii. And literally, it was a pure culture. And it was if you filtered it out or you used antibodies, it didn't work. And I'm happy to say it wasn't the one that causes infections in hospitals. But anyway, good question. It was such a shock, yeah.
I'm trying to build the bridge between what you're talking about here with the plants and the root tip and the autoimmune diseases of the human. And the issue in the human is recognition of specific moieties that are not compatible with what the body has been normally saying, these are my own cells. These are, something different, and so we've got to attack them. So it's autoimmunity. They're not self, and so they have to be hit.
Yeah.
Is there anything in any plant in which a plant has to respond to non-self cells within it? I think of crown gall and the things where viruses get into tulips and various things, but is there any model in plants where the plant cells themselves do something in a mutational way or such, and that the plant then has to attack them as bad guys?
Barry? Yeah, it is a really good question, yeah. Yes.
I'm just, I don't know, in response to pardon? Yeah, actually, so they're doing it, they're sending out the border cells, whereas humans do it within themselves. So that's where the parallel is, but the plants are doing it outside of.
I mean, in some ways it's pretty simple in terms of the fact that if you have an inflammation, that's why cancer develops from chronic inflammation, because the nets keep coming back, building up, and that's what feeds the development of the cancer. But as far as the immunity, autoimmunity, I think it's probably going to raise some questions about what we really understand about that. I mean, that's the way, because obviously the neutrophils do respond, and that's why they keep non-self. Yeah, non-self. But that's at the heart of what we have to know in order to do something about this and go beyond just describing it and say, I don't know, but again. Okay.
So back to the compost. Did you just use his compost, or did you
just Tom's compost?
Okay, so we need to get.
I think. Did we use another one, Roberto? Oh, that's right. That's right. Yeah, that's right.
One more? Yes?
You guys are too nice to come here, just to come at 4 o'clock on Friday and ask questions. I feel so
Marta, thank you. Very interesting presentation. I'm just curious about, the effect of the environment there, and how does that actually impact the formation of this with the chemistry of the environment. And have you looked into that, or are there studies on
You've hit the nail on the head of why I shifted into SWES. So I didn't have to learn all that stuff. I could ask John. But no, that's a very, very, the one thing we know is the free water is critical, and that's what we're looking at with the metals. How long does it have to be exposed to the water before it can trap the metals, and what effect does it have if it's in a clay loam soil versus a sandy soil? All of those things are going to be so critical. The simple part is you have to have free water. To grow, you have to have free water. Not to grow, 99% humidity you can grow. But in order for the border cells to come off, and the dynamic of that is so overwhelming, and you add dirt, and it's just time to retire. Yep. Yeah. All of those, they all affect that. Border cells hate pH above about 4.5. They regulate it very, very tightly. So unless you have a very strong buffer, it'll stay at that. But if you do have a strong buffer, it kills the cells. So like you say, all of those kinds of things. Yeah. 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 Yeah, there's a lot of pectin out there, and you break that down and zoop. No, actually, that was one of the things that we changed when we altered expression of the pectin esterase. The pH went up to 8, and boom. Subtle.
Primarily, that's my interest. And you know, I've read that the plant and the bacteria and fungi in the rhizosphere adjust the pH to the pH that plant needs. So when you do a pH test of your soil, it's not really that relevant.
It's really not, because it's right at
so
this.
all these agricultural people that spend millions of dollars, you know, liming their fields or doing the opposite, you know, acidifying their fields, or sort of wasting their time.
Yeah, Mother Nature's got it all under control. Rob, yes.
A question about the border cells separate from the root to produce the extracellular traps.
Here we go for a second.
Like to get it logged.
So my question concerns the border cells moving away from the root, almost like, you know, a lizard losing its tail, and the predator plays with the tail and the lizard gets away. So the pathogen is there with the border cell, or the DNA that comes out of the border cell. Are those pathogens contained, destroyed? Are they still viable? Do they still spread to infect the plant another day?
Great question, yes. Well, we found with the nematodes, for example, they're put into a quiescent state that will last for 5 days. Even if you just leave them in that liquid, they'll come back to life after 5 days. Now, at that point, they're not going to be any danger to the root, because the region of elongation is going to be gone. They have to be able to get into the meristem region to develop. So, and the zoospores, it actually does kill them. They germinate and then they're dead. The fungus, same thing. It's in a completely quiescent state, but if you take it out, put it in a nutrient medium, it'll start growing again. So that's a really great question, and it's clear that this is mainly programmed in order to protect Darwin's brainstem. And I had a brainstem injury, so I can testify that it really makes a big difference.
So I think the lively discussion proves that you have presented to us an incredibly interesting, novel, and challenging presentation that talks about the complexity of plant life. And we are used to just taking a seedling or a seed and throwing it on the ground. The knowledge that you have exposed us to shows how intricate plants really are. And thank you so much again for sharing this with us.
Thank you for working out, Rafi. It's been really fun. And I didn't burn out the microphone. Yay! Oops, did that go on? Oh, is it? Sorry.